All right, guys, so today we are going to do part 2 of cell division. In part 1, we talked about mitosis, which is the process of making body cells or somatic cells. When you make two daughter cells, they are identical to the original cells, which makes sense. If you're trying to make more skin cells, you want the new ones to be the same. If you're trying to make new heart cells, you want the new ones to be the same. So today we're going to be talking about a different process called meiosis. So how I remember this one is think me is sexy because meiosis is the process of making sex cells. So this is only for eggs if you're a girl and sperm if you're a boy. So here's meiosis. Meiosis is a type of cell division by which gametes is the fancy word for sex cells, just like somatic is the fancy word for body cells. You can remember that by thinking sex is a big game. So gametes go with sexual reproduction. It's not really. Be careful out there. Okay? So our gametes are eggs and sperm, and they are produced differently than our body cells. Because what happens is you actually have to get your number of chromosomes down to half the original number, so that when dad's sperm hits mom's egg, you get back to the original number. So in the case of humans, our chromosomal number is 46 if you're normal. So that means in mom's egg, you need 23 chromosomes so that dad's sperm has 23 chromosomes. And when they come together, you're back at 46. So they have half the number of chromosomes. So when we're looking at meiosis, how this is accomplished is you do replicate your DNA in S phase of interphase, just like you do in mitosis, but you divide twice. You only copy your DNA once, and you divide it twice. So you land up with half the number. Okay? So let's get started. And by the way, look at this picture down here. There's a picture at the bottom. That's a real human egg under a scanning electron microscope. And those little things hanging off of it are sperm cells. And the first sperm cell that gets into the egg makes a barrier after that, so the rest cannot get in. And you can tell how much larger the female egg is than the male sperm. So when we're talking about chromosomes and how they are lined up, we're going to be talking about what's called homologous chromosomes. And we've used this root word homo in the past. It means the same. So these are chromosomes that have the same size, the same shape, and the same traits on them. Okay? So we're back to that idea of alleles, that there could be a trait for hair color on this one and a trait for hair color on this one. But this one could be a brown gene and this one could be a blonde gene, okay? But they're the same size, shape, and traits. One of these you inherited from your mother, the other one from your father. And so if you look over at this picture, this is called a karyotype. We're going to be learning about karyotypes after Thanksgiving break. When you look at a karyotype, these are your chromosomes. They're actually chromatids right here, single guys, because they're not attached. One came from your mom and one from your dad. So you have 23 pairs, 2 in each. That gives us that magic 46 number. So these homologous chromosomes, as you can tell from the animation, do something very interesting called crossing over. So think of it like crossing your legs. The chromosomes cross legs, and when they do this, little chunks of each switch over. All right, so this shows one crossing over, switching up your chromosomes. And what this does is it kind of blends your genes together so that you get more variety when you split these chromosomes apart. So instead of having 2 all red ones and 2 all blue ones, you're going to have an all red one, a red one on the top, blue on the bottom, a blue on the top, red on the bottom, and a blue. So it mixes up your genes, more genetic recombination. You're recombining your genes, right, to get more variation. Because when you're talking about living organisms, variation within your population is a huge advantage. Because if you have a large variation within your population and the environment changes, odds are somebody is going to be genetically different and be able to cope with those new changes in the environment. So the real life examples of this, some people genetically for some reason are just immune to poison ivy. They don't get it. That's me. I can roll in it all day long and I will never get poison ivy. I will never get a rash. And that's because my genes, somewhere along the line, recombine and I got the right mixture that I won't get that. Okay? So variation is a strength. We should celebrate the differences between people because of that. It makes our species stronger. So here's just another picture of that crossing over when the two homologous chromosomes come near each other and switch segments. In reality, this doesn't happen just once in your chromosomes. It happens over and over and over. So you can get all kinds of combinations. And that's why families can have a bunch of kids and every kid comes out slightly unique, right? They might share some characteristics, but they're going to be different. So like if you know that family, that 19 kids and counting family that used to be on TV, they had all these kids, and if you only had 2 options for your chromosomes, it would get real boring real quick because the kids would come out almost exactly the same, and that does not happen, all right? You get all kinds of variety, and this is part of the reason why. So when we go into meiosis, what we're going to see is that meiosis is a lot like mitosis. It's almost like mitosis happening twice, okay? But there are some distinct differences that we need to pay attention, and those differences mostly are in how your chromosomes are lining up. So if we look up here in meiosis 1, so that's the first division, and the second division we're going to call it meiosis 2, okay? So we basically go through PMAT1 and then PMAT2. So it all starts with interphase, just like regular, just like mitosis, where your G1, S, and G2 phases are going to occur. So we're going to have growth, replication, preparation, just like normal. And so here, your DNA replicates and you get your chromosomes. Now, in prophase 1, same regular stuff happens. Your DNA has to coil up so it does not break. Your nucleus degrades so you can move that DNA around, and the spindle forms to attach to it to allow it to move around. The one thing different that we need to know is crossing over. This is when crossing over occurs between our homologous chromosomes, right here at the beginning, at P1, prophase 1. All right, this is when we still have homologous chromosomes next to each other for them to cross over, because after metaphase 1, we're splitting them, right? Because we split in anaphase. So they cross over, and then in metaphase 1, they line up a little bit different. They line up as homologous chromosomes. So not single file before, but in partners, okay, right next to each other. So that is different than mitosis. They call this a tetrad because tetra stands for 4. If you've ever played the game Tetris, you got to get 4 rows of blocks in order to get a Tetris. So it's 4 because remember that each chromosome is actually made out of 2 sister chromatids. So altogether you have 1, 2, 3, 4 chromatids. All right, so we have crossing over in prophase 1. Metaphase 1, the homologous chromosomes line up in the middle, which is a little different. They're just partnered up. So in anaphase, when you pull them apart, you grab onto the center of these things, you're actually going to pull them apart to separate ends, but you're going to separate the homologous chromosomes. Instead of splitting them in the middle at their centromere, you're pulling whole chromosomes back. Okay? We will split them later in the second division. Telophase is the opposite of prophase, so your DNA uncoils, your nuclei reform, your spindle degrades. And when you're talking meiosis, this happens real fast. So right now we have 2 daughter cells, each with 1 chromosome from the homologous pair, but we are not done because we are not down to half. We have to go through PMAT a second time. So here's a picture showing you the process. Here's prophase 1. It's really tiny, but there's some crossing over occurring in there. Metaphase 1, where they line up as tetrads with their little partner. Anaphase 1, where they get pulled back as whole chromosomes, and you can see the crossing over there. And then telophase 1, where they have a nuclei, and that's going to go right into prophase. All right, so now we are in the second phase of meiosis, and we are going to go through PMAT2. There's no interphase at the beginning that already happened before prophase 1. So this second division of meiosis is actually exactly the same as mitosis. The only difference is you're going to be doing it in 2 cells instead of just 1 because you already divided once and you have two daughter cells that both need to be divided again. So both cells are going to go into prophase 2 where the DNA coils so it doesn't break, the nucleus degrades so you can move it around, and the spindle forms to attach and move it around. In metaphase 2, because we already split the tetrads, we now have single chromosomes again. So they're going to line up down the middle, metaphase middle. In anaphase, because now we only have single chromosomes, those chromosomes are going to split at the centromere and be pulled back as single chromatids to either side of the cell. And then when you get to telophase, it's the opposite of prophase. So now my DNA uncoils because I'm done moving it. The nuclei reforms to keep it protected, and the spindle goes away because I'm done with it. At this point, our division is complete, and we make 4 daughter cells, because we took 2 cells and divided them again, so you're going to have 4, except they have half the number of chromosomes. So they call this haploid. So remember, diploid is when you have 2 of each kind of chromosome. Haploid is when you have half that, 1 of each kind of chromosome. So here's a diagram going through again the whole process. You start up here in interphase with your full nucleus and you have chromatids. By prophase 1, you are forming your spindles and your homologous chromosomes are going to duplicate. They're not quite there yet. Over here, the spindle is forming more. Prophase 1, crossing over is occurring and you have homologous chromosomes. Metaphase 1, they line up in the middle as partners, and you can see all that crossing over that occurred. Anaphase 1, they start getting pulled apart as entire chromosomes because you're splitting the tetrad. Towards the end of anaphase, your cell starts pinching in. Telophase, you created 2 nuclei. You're going to get rid of your spindle, and your DNA will eventually uncoil. Now, so at this point, you have cytokinesis occur. We're at splitting of cytoplasm, and you have 2 cells, but they're not done because we still have chromosomes. We go into prophase 2, so your nuclei go away, your spindle reforms, your DNA is coiled up. In metaphase 2, they were lined up single file in the middle, and then we will split them in anaphase, pulling back just chromatids. Then at the end of telophase and cytokinesis, you reform your nuclei, and you notice you have single chromatids in there, and we only have 2 per cell, and we started off with 4. So these are haploids. So when I take one of these sex cells and combine it with another, I will be back at 4 after fertilization. So here's an animation of the whole process. So right now in metaphase 1, anaphase 1, telophase 1, cytokinesis. Then we will go into prophase 1, 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. And we get our 4 daughter cells that are different now. They can't be the same because they've had crossing over occur and they have half the number of chromosomes. So our different types of cells, just to review, somatic cells are body cells, all right, which is every cell in your body except sperm and egg. Every cell in your body except sperm and egg has 46 chromosomes. This is called your diploid number because you have 2 of each kind, one from mom, one from dad. And so they represent that with a 2n, so 2 times the number of different chromosomes, okay? You have 23 pairs. And your germ cells or gametes, those are sex cells, sperm and egg. They're the only ones that are different. They get half the number of chromosomes, so they each have 23, which is haploid represented just by N. When those 2 N's come together during fertilization, you'll be back to diploid 2N, and from that point your baby divides by mitosis. So here is a karyotype, a picture of your chromosomes, of a haploid cell, which would be a sperm or an egg, where again you got 23 kinds and single chromosome in each. So that's your 23 chromosomes. When the sperm and the egg come together, you get a body cell, which is diploid, 2 of each. The pink ones are from mom, the blue ones came from dad's sperm, and you land up at a full 46 chromosomes. So there is something else that gives us variation besides crossing over, and that's called independent assortment. If you've ever bought an assortment of candy, you know that's a bag of candy that has a variety of kinds in there. Well, independent assortment is when you get a variety of genetic outcomes because your chromosomes separate independently of one another. And this also leads to more variation between you and your siblings. So if we look at the cells over here, I have the G cells, and they are two different kinds of Gs because that represents our different kinds of alleles, and my Y cells. So in this case, I have a blue on the top, a yellow on the bottom, a yellow on top, blue on the bottom. So when they separate accordingly. But how they line up here at the beginning during metaphase is totally random and not related to one another. So you can see the same genes in a cell down here that arrange themselves a little differently at the beginning, and so they divide differently, and the end results, the combinations you get at the end, are also different. So that's independent assortment, which just gives us more variation. And remember, variation is a big plus when you're talking about a species. So now let's get specific into females and males. Women do this process called oogenesis. That's when we go through meiosis to make egg cells or an ovum. So it's a really funny name, oogenesis. Ovum is a fancy word for an egg. When women produce their egg, they produce 4 cells, but 3 of them are duds, and one is this big giant cell. We only get one good egg produced by meiosis. And the other 3 things are called polar bodies, and they just disintegrate. And the reason why this happens is because that one big cell gets all of the organelles in the cytoplasm from the other 3, because it is the female egg that supports the development of the fetus. The male sperm is really just there to transport DNA. It's not giving any of the organelles or even the cytoplasm, just DNA. So you can think of it this way as the female egg is expensive and rare, all right? Women release one of these eggs a month on average, and they only do it for a short period of time, from the time you go through puberty until you go through menopause. Now, men are different, and we'll get to them. So here is a diagram showing the process of oogenesis. This is your immature egg. So you guys had your eggs while you were still a little fetus developing in your mom's uterus. You already had your eggs in there, immature, just waiting in your ovaries. So these eggs have been exposed to everything you have been exposed to. So you really need to think about the choices you make in life, because those choices can affect the cells inside your body, including these little resting eggs in here that are in your ovaries. So for what mother's life choices are really important to the development of a fetus. So when you go through puberty, you start shedding one of these eggs. It usually happens about the middle of your menstrual cycle, not when you're actually having your period, but in the middle. And your hormones spike and you release an egg, and some women experience a cramp at that time because it really is bursting out of the ovary. When it does that, it has already started to divide, right, into a primary oocyte. And during the first division, which would be PMAT1 or meiosis 1, you can see the unequal distribution of the cytoplasm, which is where all your organelles are. So this little polar body is going to divide again, but it's going to make these tiny little polar bodies. And this guy is going to divide again, but it's going to be unequal again. So you do produce 4 cells, but only 1 is actually functional. And these other 3 polar bodies, your body just reabsorbs them. So rare and expensive to make is what the female sex cell is. Now for the boys, the process of you producing sperm is called spermatogenesis. So when you make sperm, you make 4 fully functional sperm every time, and you're constantly making them. It's kind of like a conveyor belt of sperm. And boys make sperm from the time they go through puberty until the day they die. Okay, now the quality of the sperm might go down, but you will still be producing them. All right, so very different than women. So where women's eggs are rare and expensive, boys' sperm is cheap and easy to make. So here is the process at the bottom of making sperm. It is just regular old meiosis, where you copy your DNA, you divide once through PMAT1, you divide twice through PMAT2, and then you have all your sperm. It takes 4 months to produce a sperm cell in your little conveyor belt, but you're constantly making them. So as soon as some are released, there's another backup just waiting there. But what that does mean is for the time when you are making children, when you decide to have a child, and hopefully you purposely decide that, you need to have some clean living at a minimum 4 months before you make that child, so that those sperm are the best quality sperm you can produce. That way you're giving your child the best start they can have. Unlike mom, who has to be careful for her whole life, 4 months is usually pretty good. You might want to stretch it to 5 to make sure. And then you got some new sperm coming out of the chute. That's not to say if you don't do some extreme things to your body that it couldn't affect your fertility. It certainly could. So now let's talk about bacteria. Bacteria divide as well, and they do it in a way simpler way than we do. It's a fancy name, binary fission, but the process is simple because they don't have a nucleus to deal with. Like our cells have to get rid of that nucleus and move stuff around. Bacteria, because they're prokaryotic, they have no nucleus, no membrane-bound organelles. So they simply copy their DNA and then pinch off their cytoplasm, and that's it. So this is asexual reproduction. Bacteria, because they're only one cell, are not producing sperm and egg. You'd have to be multicellular to do that. So instead, they just copy their own DNA and then divide their cytoplasm, and they create an exact clone of themselves. So each organism gets one copy of the DNA. Now, the downside of this is they're missing some of that genetic variation that sexually reproducing organisms have. And remember, that variation is a huge strength in a species. So these guys don't have all that variation, and so you can get medicines that will wipe them out. That being said, there are mutations that occur readily in bacteria that can give them other variations, things that would make them resistant to our drugs. And we'll talk about that more later on in the year. So with all this stuff about our cells dividing and what happens with our DNA, we need to be knowledgeable about when we reproduce, what can we do to make sure that we are producing viable sperm and offspring and all of that stuff. If you're worried about your genetics, if you have a history of a genetic disorder in your family, there are some things you can do. They have carrier screening. So for example, when myself and Mr. Elledge decided to have a child, I have an aunt and uncle who have muscular dystrophy. So when we told our doctor this, they sent us to a geneticist so that we could look at our family tree, which I kind of already knew as a biologist, but looked at our family tree and said how likely we would be to have a child with muscular dystrophy, because we did not particularly want to do that. It shortens their lifespan. It's a hard life to live. But it turns out that we were highly unlikely to have that. So it worked out really well. But if you're concerned about something like that, you could go to a geneticist and they could screen if you're a carrier for the disorder. You can also have prenatal testing, so they can test your genes while you're pregnant. And this has gotten really easy in recent years, as much as just a blood test for some of these things, for things like Down syndrome. And that way you can be prepared when you have your child, like if there's equipment you need to get, or if you need to educate yourself to know how to deal with your child, you would know ahead of time what is coming instead of being surprised on the day they come into the world. Now, you could also have newborn and adult screening at any time. If you think you might have a genetic disorder, they could always take DNA out of any of your cells and run it and check for chromosomal disorders. So one of the disorders that they check you for right away as soon as you're born is this one, PKU, or Phenylketonuria. The reason why, if you were born in a hospital, every single one of you are tested for this one is because it's totally preventable, which is nice. So you're born lacking an enzyme. Remember, enzymes are proteins that speed up chemical reactions. They help break things down. And what this enzyme helps break down is a protein. And the proteins it breaks down are the amino acids, phenylalanine and tyrosine. And so, you know that saying that too much of a good thing turns into a bad thing? That's kind of what happens here. Baby is born normal. Everything's going fine. But these amino acids are found in milk products, formula and milk. And so as you start feeding your baby, they start building up toxic levels of these amino acids in their body. And what that does is it lands up damaging brain tissue. And your baby can go from being perfectly normal to being mentally incapacitated and start having seizures, and it's really awful. So all you have to do, if you know your baby has it, and it is incredibly rare, it's not a common thing, but if your baby has it, you just have to feed them a special diet. And so they have formula for babies who have PKU. And what this will do is prevent them from being mentally handicapped just by feeding them a different diet. So even though it's incredibly rare, it's one of the things when they stick the baby and they take their blood right away after birth that they go ahead and test for so that they can prevent that from happening. So hopefully you guys know a little bit more about how your cells divide, and I will talk to you next time. Bye.